see the F plasmid is integrating. Here, the F factor now has inserted itself into the donor's chromosome. So now we have our HFR cell. And remember, HFR stands for high frequency recombination. Now we're going to take this donor and mate him with an F negative recipient and see what happens. So here's our mating, our HFR donor, our F negative recipient. And what's important for us to be aware of is when the gene transfer from the male to the female occurs, it always begins about the midpoint of the F factor. It's called the origin of transfer. So what happens is the donor initially starts to transfer half of the F factor to the female, and then what follows is a copy of the donor's chromosome. And an entire copy of the chromosome must be transferred until the last half of the F factor is transferred to the male. Okay? So we see, and it's actually kind of hard to see here, this would be the first half of the F factor, and now it's being followed by a copy of the male's chromosome. Now, for the entire F factor to be transferred to the female, an entire copy of the male chromosome also must be transferred. And this takes a long time, maybe 60 to 90 minutes. And if you can imagine, you guys, this happening, say, in your intestines, with the movement of all the food and feces and fluid, it's easy to understand why those matings are often interrupted. Literally, the mating bridge breaks. So the mating is interrupted. And that means in nature, the recipient usually only receives half of the F factor. And a copy of some of the donor's chromosomal DNA. Okay, so with what we see here, after we have an interrupted mating, the donor remains HFR, okay, so he remains a donor, a male, but because the recipient only got half the F factor, she remains a recipient, she remains female. But what she may have gained was a homologous sequence of DNA from the donor, which through homologous recombination, she can insert into her chromosome. And this is where this conjugation becomes important. Here is another way that donors can transfer copies of antibiotic resistance genes to their neighbors. They can transfer copies of toxin genes to their neighbors. They can transfer copies of, say, capsule genes to their neighbors. So this is why conjugation is so important to us as humans. Again, another way for new antibiotic resistance pathogens to evolve, or a way for new bacterial pathogens to evolve. Yeah.
Okay, so you said that with all the movement, the bridges break. Is it safe to say that the more physically active a human being is, you decrease this activity in your assessment?
Okay, gosh, that's a great question. Yeah, exercise and staying regular, like eating lots of fiber, because you want to keep things moving. I think the one problem is that this initial transfer is probably pretty quick. So you're absolutely right. Let's say you have a really active digestive tract and you yourself are really active and there's a lot of movement going on, you would reduce the chance of a successful mating in that both parts of the F plasmid would not be transferred. So you decrease the chance of converting more of your bacteria into donors, which we don't want. We don't want donors, right? We don't want more and more donors sharing more and more genes. So absolutely, by remaining active, we could argue, yeah, you would reduce the chance of these successful matings occurring. Successful meaning that the recipient gets converted into an F plus donor. But I think the problem still is those males that we do have, you're probably still going to be successful in getting at least some of their chromosomal genes, copies of the chromosomal genes from the recipients. But you're right, anything that would help interrupt this mating from a human point of view is good for us, especially, yeah, and oh my gosh, that's such a good point. Because in hospitals, what do we do when we're patients in hospitals? Yeah, that's mating heaven for our bacteria, right? Yeah, so I think that is such a cool thought. Yeah, that physical activity, you could argue, is going to help decrease conversion of females to males, and that is definitely going to decrease overall horizontal gene transfer of bacteria. Yes. Mm-hmm. Yeah, that's good. Yeah, and don't you often think about, you guys, hospitals when people are sick, and obviously, they're sick, they can't get, but just think, maybe therapeutic massage, you know, some even where they don't have to get up, but just maybe some abdominal massage, just really keep things moving, muscle massage, all of that I think would be really helpful if the patient, you know, wasn't burned or really bad injury or sore. Yeah. Okay. Yeah.
Can that negative cell, the one that got part of the donor's DNA, can it get another donor come in?
It can, yeah. Oh, great. Yes, absolutely, guys. This baby remains female. She remains a recipient. So there could be multiple males that will continue to mate with her as long as she remains F negative. Yeah. Yeah. Yeah, bad, huh? That's not good. Yeah. Okay. Okay, promiscuous, they are promiscuous. Okay, so we're going to move on, you guys, to the last example, horizontal gene transfer. And this process, you guys, is really complicated. To really understand transduction, which involves bacterial viruses, we really have to understand bacteriophage replication, which is fascinating but very complicated. So what we're going to do for right now is just keep it real simple. And then what we're going to do is, the next unit we're going to do is chapter 13, viruses. We're going to go into great detail on how bacterial viruses replicate. And that's what this cartoon is for on the front sheet of your handout. So this cartoon will be useful to understand transduction, but also be really important on Thursday when we go through all the steps of bacteriophage replication. So we're going to keep it simple for right now. So transduction is when donor bacterial DNA is transferred to a recipient by a bacterial virus, the bacteriophage. Now the name phage means to eat or eater, and historically when the activities of these bacterial viruses were first identified, it looked to microbiologists like the bacteria were being eaten, and hence the name bacteriophage. Okay? And we now know these little bacteria eaters are actually viruses. But we often use the term bacteriophage or phage to describe them. Now, this cartoon, I know it's not very big. This is figure 8.28 in your book, and this is showing a phenomenon called generalized transduction, when any random piece of a donor bacterium can be transferred to recipient. And we start this process by looking at this would be our donor bacterium. This little turquoise kind of lunar lander looking thing is our bacterial virus. The turquoise portion represents a protective protein coat called the capsid or the head. And the capsid or the head protects the phage DNA, which is here is kind of this pink ribbon color. Now what happens is the bacteriophage attaches to specific receptors on the surface of the bacterium, and it literally injects its phage DNA into the donor bacterium. Within the cytoplasm, unfortunately, the bacterial RNA polymerase transcribes the phage genes. The bacterial ribosomes translate the phage mRNA to make phage enzymes and proteins. And what happens at that point then is that the phage basically take over the bacterium and convert it into a virus-producing factory. That's a great description Professor Meyer uses. And as part of converting the bacterium into a virus-producing factory, part of the phage enzymes, they hydrolyze the bacterial donor chromosome into these little fragments. And the reason is the phage are going to use the bacterial nucleotides to make more copies of the phage DNA. So we see the phage DNA is being copied here. Now, after we get phage DNA copied, after we get phage proteins made, the proteins self-assemble into these capsids. And this is such a cool event, you guys. The capsids then will bind to and insert the phage DNA into the capsids here. So we can see that we're getting assembly of a complete phage here, 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 here. Whoops, what happened there? What happened there? By mistake, a piece of the donor chromosomal DNA got packaged, because to these phage heads, DNA is DNA. And the phage heads can't tell the difference between phage DNA sequences and bacterial DNA sequences. So occasionally a mistake is made and a piece of bacterial donor DNA is packaged into the phage. Now this is a mistake, and these little mistaken phage are called transducing phage. Okay. Now when this bacterium is killed as a consequence of the phage replication, the bacterium lyses, it releases all the phage, even the transducing phage. And these transducing phage are still infectious. They can still bind to a neighboring bacterium, the recipient. They can inject the DNA. In this case, it's donor chromosomal DNA. And through homologous recombination, that donor chromosomal DNA may be inserted into the recipient's chromosome. So we can see the horizontal gene transfer occurred by a bacteriophage or virus. So this is called generalized transduction. Again, because any random fragment of the donor can be transferred to the recipient. So here we just see the donor DNA that's been inserted in the chromosome, and it can remain there forever and just become part of the recipient's chromosome. Now, when we actually get into chapter 13 and we talk about, there's two different ways that bacteriophages can replicate themselves. There's another cycle that permits phage to carry out a different type of transduction called specialized transduction. But we won't describe specialized transduction until we get into chapter 13 and we talk about phage replication. All right, so again, you guys, just in summary, that was 3 different ways that bacteria can exchange genes horizontally. Transformation, uptake of naked DNA by competent bacteria, conjugation, transfer of donor DNA to a recipient that involves cell-to-cell contact, and transduction, in which donor bacterial DNA is transferred to the recipient by a bacterial virus or bacteriophage. Okay. All right. So, woo, I almost forgot. Oh, this is great stuff. Okay. So we want to ask ourselves again, why is this transduction, important? Why do we care? That's a real important question to always ask yourselves. Why do we care? Why is this important? So let's just back up, you guys, and we want to address this question that is from the textbook, and that is, how can transduction contribute to the emergence of new bacterial pathogens? And this is a real hot topic of great importance here in California. So we want to just end the lecture with this discussion of transduction and transfer of bacterial toxin genes. And the example we're going to use is the evolution, the emergence of a toxin-producing E. coli. Actually, two different strains. And these are toxin-producing strains of E. coli described as O157H7. And just last May in Germany, a new strain, toxin-producing strain, has emerged called E. coli O104H4. So first, you guys, let's just review what the O and the H represent. And hopefully this will ring a bell from Chapter 9 Motility in Lab. So you guys, what is that H antigen? You remember it's German for Haut or breath. So what part of the bacterium is the H antigen? Excellent, you guys. This is the Flagellar Antigen. Remember, flagella are made out of proteins, and proteins are excellent antigens. Antigens are substances which trigger an immune response, such as antibody production. So if we're infected by a bacterium that has a flagellum, our bodies will make antibodies against the flagellum. And we can use those antibodies to identify specific strains of bacteria in a process called serotyping. Use of antibodies to identify specific microbes.
Is that a goat?
Dynamite, guys. Okay.
It's a goat. It's a soda.
So what about the O antigen? What does the O antigen represent? Remember, in German, it stands for ohne Haut, without breath. Do you remember what the O antigen is?
Without flagella? Or
And remember, guys, it's so good to guess. Don't be afraid to guess. That's really how you learn is by guessing. So ohne Haut, no breath, no motility. You find the O antigen in gram-negative bacteria. It's part of the unique outer membrane. What's a unique component of the outer membrane? LPS, dynamite, you guys. Okay, so the O antigen is the polysaccharide, the chain of sugars. Lipopolysaccharide, and you want to remember that's also, it's toxic, so we'll call it the endotoxin. Okay, and again, now polysaccharides, they aren't the best antigens, but you can get low-level antibodies produced against different polysaccharides. So the polysaccharide portion of lipopolysaccharide will trigger antibody production. And what we want to see here, you guys, is that there's many different O antigens. I mean, there must be at least 157 of them, right? Because this particular toxin strain of E. coli is O157. The newer version, the newer toxin-producing version is O104. And we can see up here there must be at least 7 different Flagellar antigens, because this one's called H7, the newer toxin-producing strain is H4. Okay, so just be aware that these O and H antigens are really important in tracking disease-causing strains of E. coli, right? Many different strains of E. coli. Most of them aren't harmful like the ones living in our intestine right now, but some of them produce toxins, can cause horrendous foodborne outbreaks. So we need to be able to track exactly which serotype we're working with. Okay, so why the heck are we talking about this? Well, it turns out that prior to World War II, E. coli O157H7 wasn't even considered a pathogen. It didn't make toxin. What the heck? What happened? So what microbiologists believe is that a gene for a powerful toxin, one of the most powerful bacterial toxins, called shigatoxin, the gene for this powerful bacterial toxin, shigatoxin, was transferred from a close cousin of E. coli called Shigella dysentery that caused a maxillary dysentery. A bacteriophage transferred the shigatoxin gene from Shigella dysentery to E. coli O157H7. And remember, the idea is this guy previously didn't have a toxin gene. It wasn't a pathogen. It wasn't until a bacteriophage carried a copy of the shigatoxin gene and inserted it in E. coli that E. coli acquired the shigatoxin gene. So this was through transduction. So now we've got an E. coli that now can produce this shigatoxin. And this shigatoxin, you guys, is so powerful. I think I read where it's next to botulinum toxin. It's one of the most powerful toxins of bacterial origin. So what happens is if we eat food contaminated with E. coli O157H7, okay, and this happens often because cattle can carry it, but they don't have any signs, so they're often asymptomatic. Because one hypothesis is their cells lack the receptor for the shigatoxin, so the E. coli can grow in their intestines, and that means it's excreted in their feces, right, in their manure. So wherever the manure goes, the shigatoxin-producing E. coli goes. This E. coli O157 initially emerged in the 80s as a so-called Hamburger Pathogen. Well, what happened is that when the cattle were sent to slaughter, feces would get on the carcass, and in the feces would be the E. coli O157H7. And what happens is when beef is contaminated with fecal pathogens on the outside, if you grind it up, the fecal pathogens get inside the ground meat. So that if you don't cook it thoroughly, the fecal pathogens are still alive in the center of your hamburger patty, right? So that's why we associate ground beef with this. So what happens is, and this was heartbreaking, children are really very susceptible. If the kids ate these hamburgers that were contaminated with E. coli O157H7, E. coli started growing in their intestines, they started producing shigatoxin. Shigatoxin shuts down, it inhibits eukaryotic protein synthesis, so it inhibits ADS ribosomes. And it can have a lot of damage. It can cause a bloody diarrhea. Yeah. So that's often one of the first signs, although not all children develop bloody diarrhea. And in a small percentage of the kids that develop these infections, the shigatoxin is absorbed into the bloodstream, and it damages the endothelial cells of blood vessels. The vessels, and this will trigger lysis of red blood cells. So a hemolytic anemia can result, and what's even worse is the shigatoxin can circulate to the kidneys. And you guys that have had AMP, your kidneys are just this massive tiny capillaries, massive tiny blood vessels, and it can cause severe kidney damage and failure. And as a result, you get an increase in urea in the blood. And so you get a uremia. together then are described as HUS, hemolytic uremic syndrome. And it can be lethal, it can kill little children. Okay, so we're at time right now, you guys. So we'll pick up here just to talk a little bit more about the Shiga toxin producing E. coli and what we can do to prevent infections. And then we're going to move on to just the last part of chapter 8. We're going to look at antibiotic resistance and natural selection for antibiotic resistant bacteria. And then we're going to watch. We're going to assign 2 topics in chapter 9. PCR and gene cloning as homework assignments so that we can jump right into the chapter dealing with viruses. Okay? And I will put a little worksheet, you guys, for the chapter 9 homework topics on D2L.